Okay, so you should be able to see my screen and laser pointer.
That's great. Could you just
Good.
forward one slide? Move, just move one slide. Okay.
That is my next slide, I
Okay.
promise. It looks weird, but yes, I always thank people that did the work first. I know it's a little
Well,
strange, but
that's the most important slide, right?
yeah. Yes, no, absolutely. 100%. And that's why I always put it my second slide. So if I run out of time or something happens, the people who really did the work,
Yeah,
um, you
exactly,
know,
exactly,
it's,
exactly. Yeah.
yeah.
So you do
Cool.
lots of imaging, right? So
Yeah,
for
I'm also director of our imaging facility, or
oh
one of the
oh
imaging facilities here.
oh
So yeah, we do things like
wonderful.
light sheet, intravital, super resolution. I don't, but our microscopes do things like FLIM and FRET and all kinds of other fun
Oh
stuff like
wow.
that. Yeah. So
Well,
yeah.
that's very comprehensive. So from the tissue level to the single molecule level, that you can all get covered.
Yeah, no, it's try to build the core out that way and to complement other cores on our campus. So we don't directly compete with other cores in terms of, I
Uh-huh.
mean, basic confocal we do, right? Because everyone is
Yes, yeah,
does
yeah.
basic confocal. But yeah, so we try and actually make sure we're, complementary and so that we have lots of different cool pieces of tech and everyone can do stuff they need. So yeah.
That's awesome. That's awesome. So that's shared among the entire Davis.
Yeah, whole university.
Yeah,
Yeah, absolutely. So yeah, it's been fun. A lot of work, but fun.
yeah. I also like imaging a lot. Of course, in the past we did a lot of so trying to look at how T cell dynamically interacting with the
hmm
presenting cells. So using turf, using
hmm
focal, and also using light sheets. I think that's pretty. I would say so every time when I watch a movie, that can make my day. A good, really
Yeah.
good movie.
Yeah. Yeah. Are you guys doing lattice light sheet as well for the like dendritic cell
Yeah.
T cell
So
interaction kind of stuff or target?
yeah, we have a supporting person here at Yale, Shaolin, who used to work in Genelia Farm.
Oh, wow. Nice.
Yeah, so he actually set up the system, and then we can
Awesome.
work on on the T cells interacting with the antigen presenting cells or a cancer cell. So
Yeah.
that's pretty
Super
cool.
cool.
Yes.
Yeah, absolutely.
Okay. So Colin, usually we started on time.
Yep. All
Is
right.
it
I'm good.
okay if we just get started?
Absolutely. Yeah. Let's go
Okay.
for it.
Okay. Good. All right. Um, Welcome to today's Immune Zoom seminar. It is my absolute pleasure to
Recording in progress.
introduce today's speaker, Dr. Colin Riordan from UC Davis. Colin obtained his bachelor degree in McMaster University near Toronto and a PhD at the University of Calgary. He received a postdoc training in the Princess Margaret Hospital in Toronto and the La Jolla Institute of Allergy and Immunology in San Diego. In 2014, and Colin started his own group at UC Davis and was promoted to tenured associate professor in 2020. The primary research focus of Colin's lab is the control of innate and adaptive immune cell function by the nervous system. With training focused on mucosal immunology and inflammation, he has developed a research program to dissect the functional neural circuitry that allows for the control of immune cells activation systematically and in both the intestine and lung, and understand the contribution of neural immune interactions during bacterial infections. So as I was just chatting with Colin, he love microscopy, love imaging. So I think in his talk we will see a lot of beautiful images or movies. So without further ado, Colin, the floor is yours.
Awesome. Thank you very much for the kind introduction and the invitation to speak here. And of course, everyone for joining us, no matter where you are in the world or what time it is there. And so I'm going to first start by thanking all the people who actually did the work in the lab. So talented graduate students and postdocs. my core facility director, and then various collaborators, and of course, NIH for funding this work. So I'm going to talk to you today about neuroimmune circuits. And as was mentioned, my lab basically looks at a few different things, how the nervous system interacts with the immune system and how immunological outcomes could be modulated by the nervous system and vice versa, right? So how the immune system could signal back onto the nervous system. But first, I thought maybe, or first of all, what I'm going to talk about today, though, is a limited discussion intestinal inflammation. And I think any discussion on neuroimmune circuits should probably start at the more recent beginning. And so this is systemic inflammation, where a lot of this work came back into play, or became more popular again and really started being investigated now that we had newer and better tools compared to the 70s and 80s. And so this is really, I think, 30 odd years of the work of people like Dr. Kevin Tracy and his group, and many, many researchers within his group. And this is the cholinergic anti-inflammatory pathway. And so when I talk about neuroimmunology, I'm talking about bidirectional communication between neurons and immune cells, right? There's neuroimmunology is a huge field and it means lots of things to lots of people, but my group is interested in the periphery. And this cholinergic anti-inflammatory pathway, or CAPE, was originally identified by Kevin Tracy and his group as a reflex arc. And quite simply, and again, this is an oversimplification, but I think illustrates the point. There's the idea that neurons, specialized neurons, vagal afferents in this particular case, are capable of sensing inflammation. So either cytokines directly, or they have the potential as well to sense damage-associated molecular patterns, or others have shown maybe even directly constituent molecules from different pathogens, like pathogen-associated molecular patterns. This information gets interrogated in brainstem and discrete brain loci, and there's an efferent signal that's sent out. There's a motor signal that takes us, or is the consequence of this coordination of information, and is sent out to the body in the periphery, where in this particular case, the signal is going to the spleen and these sympathetic neurons that make catecholamines. If you recall from maybe early days of studying physiology, these sympathetic neurons making catecholamines such as norepinephrine. Release of norepinephrine in secondary lymphoid organs such as the spleen activate a specialized population of T cells. These are cholinergic acetylcholine transferase positive T cells. This is the enzyme that makes acetylcholine, and that's abbreviated as CHAT in future slides. And so this enzyme CHAT makes acetylcholine, which we think of as traditionally a neurotransmitter. And here, though, what the cells are, the T cells are doing, the specialized subpopulation of T cells do, is release acetylcholine under these very specific conditions, receiving norepinephrine. Signal gets interrogated on the cellular level by beta-2 adrenergic receptor, and this can suppress NF-kappa B signaling in macrophages, blocking TNF-alpha production. At least in models of sepsis, this is the predominant effect that happens. Now, as I'm sure many of you are aware, NF-kappa B is central to immune cell activation, and in fact, aberrant NF-kappa B dependent TNF-alpha production occurs in many different diseases. And two diseases of interest in these infographics from their particular patient advocacy groups. You're not supposed to necessarily read through all of this, but it just highlights how many people, at least in America, are suffering from these disorders and how much this actually costs the economy, right? So there's a healthcare cost, there's a cost to, and beyond just even that, there's just the quality of life that is significantly reduced for these people. And while we have many different medications, and including anti-TNFs that have been developed over many years and are life-changing for some of these patients, they don't work for everybody. And over time, they can lose efficacy. So there's still been a strong drive to be able to control aberrant or inappropriate immune activation. Now, all of this information has led up to these open label clinical trials of neural stimulation devices. So it doesn't just have to be a reflex that stimulates this efferent outflow to turn on specialized immune cell populations to turn off aberrant immune activity and prevent inflammation. we can also stimulate this electrically. We can activate nerves specifically to actually drive a protective anti-inflammatory effect. And this is work, again, by many, many different people, including Dr. Tracy's group and various companies that I'm showing you here, and they're different devices, or at least at one point, they're different devices. Now, this is a rapidly changing space, and this is the only part I'm going to touch upon. So there's already a lot known about neuroimmune circuits. It controls some aspects of inflammation. But what do we know about the intestinal tract and intestinal inflammation and the role of neuroimmune circuits there? And I would argue not very much. Now, what I'm going to start to show you are little vignettes of research that my lab has published, and end with currently unpublished work that we're very close to submitting. So you'll have some unpublished data to see. And what I would like to propose to you, or have everyone start thinking about it, is that there's many different neuroimmune circuits that can communicate between immune cells and nerves, but that we can also repurpose these cells, or these cells can be repurposed rather, for different effects. So just because we discovered CHAP-positive B cell or T cells and having a protective role in endotoxemia, that may not be their only role. And in fact, this is true. And so when we were starting to think about what happens in enteric bacterial infection, for example, colleagues of ours, so I've known Wouter de Jonge for quite some time, as well as these people here at McMaster, Elena and Chemik, they started looking at, well, what about these CHAP-positive T cells and regulating other functions? And especially in the gut, they showed that that these cells contributed to regulating the microbiome, or at least changing the microbiome. And this led us to start thinking about, what about enteric bacterial infection? So how we do this is use a mouse model. This is Citrobacter rodentium. It's adapted. It's an attaching and effacing pathogen. So it's a bacteria that, when ingested, it normally stays in the lumen, but it attaches to intestinal epithelial cells, and it does this by effacing microvilli and then forming a very, very strong attachment to those epithelial cells, where it injects a factor, bacterial effector proteins, to enhance the proliferation, survival, of the bacteria. These virulence mechanisms are very similar to human pathogens: enteropathogenic E. coli, enterohemorrhagic E. coli. And nicely, because this model has been around for so long, it's a very well characterized course of disease. In our particular vivarium, this model looks like this. So we would infect mice on day 0. These are adult mice that we're infecting. And we gavage either Citrobacter rodentium or the LB broth. So that's what the bug is grown in, as our vehicle control. And then you can euthanize the animals at different time points and take tissues and do all kinds of different things. So you can look at histopathology. This is typically a colonic hyperplasia, so the intestinal epithelial cells are rapidly proliferating. You can do things, of course, like look at the bacterial burden by enumerating the number of bacteria that are either in the feces or in the tissue, or if there's inappropriate or bacterial translocation to other organs. And of course, you can look at post-immune responses in many different ways, qPCR, flow, and that sort of thing. So this is the model that we like to use in my group. And I'm going to show you just this first vignette here, that CHAP-positive T cells are not just for control of endotoxemia, inappropriate responses during endotoxemia, but they're also important for host protection during enteric bacterial infection. So on the left here, we're just seeing our wild type. These are all, these are CHAP GFP mice, so they express GFP under the CHAP promoter. So any cell that's going to make acetylcholine because of having CHAP is also going to be marked by GFP. And you can see this is in the colon of a LB gavage mouse. So this is our control non-infected mouse. You can see there's green fibers here. These are actually nerves. And you can see here's a tuft cell. And you can also spot, if you look very, very closely, there's some T cells that are, so red are the T cells, with green are the GFP. So that would be a CHAP-positive T cell. If you infect with Citrobacter rodentium, though, what you can see is there's a lot more T cells, as you would expect. This has been known for quite some time. But there are also T cells that are CHAP-positive. And this is quantified here over a couple of days. So day 6 post-infection, day 10, That tends to be the height of the infection in our colony. And this persists out to day 30. This is typically where clearance is. These cells are actually important. So we have the ability to make a T cell conditional knockout. So we have chat flox mice, cross them with LCK Cree. And in those conditional knockout mice, you can see there's in the colon a significant increase in the amount of Citrobacter that's found there. So their bacterial burden is significantly higher. If you look by confocal microscopy and you stain for epithelial cells in green, so these are uninfected mice. Here's our wild type here, our chat T cell conditional knockout mice. There's obviously no Citrobacter in the uninfected mice. That would be in red. Now looking at our infected animals, you can see there's Citrobacter and it stays in the lumen. This is our wild type mouse. And in our knockout, there's Citrobacter there as well. But I think you can appreciate that this Citrobacter goes much further down into the crypts, that there's a change in how it's relating to the host. And so what we go on to show further in this paper here, this is now from 2019 and plus pathogens, is that there's critical roles of these chat positive T cells to aid host protective immunity. So again, we can discover neuroimmune circuits and components of this circuitry, but evolutionarily, threats may have arisen that these cells could function in different capacities as well. So we shouldn't just be locked into this cell only works in this particular way, that there's much more nuance in biology. Well, after this, we started to wonder, how is it that signaling could happen? And of course, we come back to Kevin Tracy's, the summary image of many works, including by Kevin Tracy's lab. And we started thinking about, well, what about sensory neurons? Are they actually important in helping to modulate downstream effects and coordinate the immune response to these enteric bacterial pathogens? This was published in 2020, and I'm going to walk you through a little bit. And of course, there's been many people, it's not just our group, that have come up with this or have shown similar effects. Isaac Chow's lab also showed in Cell in 2020 using Salmonella that in fact sensory neurons are important there as well. And so we thought sensory neurons might be important because to activate these, and especially sensory nociceptors, right? So nerves that are going to detect noxious stimuli, they have unique signal or unique receptors to detect these signals. One of these unique receptors is this one here. It's a polymodal receptor, so it responds to lots of different kinds of ligands and noxious substances or extreme heat, for example. And it's known as TRPV1, right? And it's responsive to, I'm showing you here, capsaicin, heat, immune products, and LPS have been identified as ligands or ways that this channel can actually become activated. And so our hypothesis then was that TRPV1 positive neurons and the neurotransmitters that are released from these neurons coordinate those host protective responses during enterobacterial infection. So we're going to use our Citrobacter rodentia model again. You've seen this part before, but now to ablate those TRPV1 positive neurons, what we're going to do is pre-treat those animals and do a chemical ablation of those nerves. And we're going to use this compound called respinerotoxin. That's abbreviated from now on to RTX in the slides. And it's derived from a cactus, and that's why I have this picture here. And it's excitotoxic. And essentially, it is such a good agonist at TRPV1 that it locks the channel open and the nerves die. So you have selective depletion of TRPV1 positive neurons in animals that receive a treatment of this drug, but obviously not vehicle. And then after a week after the last dose, we come back and we either infect the animal or give them LB broth as our control, and we go ahead and look at our typical parameters. So looking for immunopathology, bacterial burden, or markers of host response. And so the ablation of these TRPV1 positive neurons with RTX, here we're just looking at our fecal bacterial burden. You can see in the animals that have RTX-mediated ablation, we start to see separation of these curves. So they start having significantly more bacterial burden starting at day 10, and this persists out at least until day 17. But we also have a delayed ability to clear these particular, or to clear this pathogen here. If we look in the colon now, so colonic associated bacteria, we can see again in the RTX. So TRPV1 nerves are ablated in these animals. There's a significant increase in the number of Citrobacter that are associated there. And if we now look at our histopathology, so we're looking at the length of the crypts. So the intestinal epithelial cells are proliferating in response to the immune response, actually. So T cells drive this increased proliferation, and that makes these from sort of the base of the crypt to the top of the crypt, that becomes much longer. And so that's what we're seeing here. So our infected animal, as we expected, yes, there's actually crypt hyperplasia, that this distance has become increased compared to uninfected. And this is further increased in our TRPV1 knockout, our TRPV1 depleted animals. So there's increased colonic hyperplasia, crypt hyperplasia as well. So not just increased bacteria, there's actual immunopathology that's changing. The reason for this, so we started looking at T cells because it is such a prominent member of the immune response, or prominent portion of immune response to Citrobacter rodentium. And so we started looking both by flow cytometry, and I'm just going to show you the confocal. And you can see that Citrobacter rodentium induces, so this is 10 days post-infection, we can see significant increase in the number of T cells that are recruited, and resveratrol, so TRPV1 depletion or ablation of those nerves reduces this recruitment. But then there's this catch-up period, and you can see this, I think, fairly well, depending on how the picture is projecting for you. Again, here's our RTX treated animals. We don't have a lot of CD3 positive T cells that have come in compared to our vehicle treated. And then at day 29, we can see there's still some here, but there's this catch-up sort of period that's happening. We started asking what could happen to drive this effect, what could reduce or be one of the reasons that there's reduced T cell recruitment at this time point. And so here we're just showing you qPCR of whole colonic tissue, and we can see that the expression of this adhesion molecule, MAdCAM1, so this would be expressed by endothelial cells and would allow for binding by alpha-4 beta-7 bearing T cells to actually allow them to gain entry into the colon. And you can see this expression is significantly reduced at day 10 and then increases significantly at day 29 post-infection. Again, mirroring what's happening here with our actual T cell numbers. And again, we've gone through and done this by flow as well, and you can look this paper up as well to see more of that data. Well, the loss of TRPV1 or ablation of those neurons is doing more than just simply losing TRPV1 expression. That we're actually destroying those neurons, killing those neurons off, and we're also, as a consequence, removing sources of neuropeptides that are stored within. And so my postdoc, my PhD student, Michael Kremen, decided that he was interested in what would happen if we had deficiency of TRPV1. So just trying to isolate what happens exclusively through TRPV1. And so what he did is use a TRPV1 knockout mouse, and just showing you day 10, looking at feces or in the colon, you can see, so again, we have, mice get infected, wild-type mice get infected, and this infection seems to be worse. There's more bacterial shedding here in the feces of the TRPV1 knockout mouse compared to our wild-type mouse. And if you look at the colon-associated amounts of Citrobacter rodentium, again, the TRPV1 knockout mouse is showing significant increases in the number of bacteria there. Looking at the immunopathology, we can see that there's already an interesting, so this is non-infected wild-type, non-infected TRPV1 mice. We can see there's already a difference in the amount of, or the depth of those crypts. So there's already crypt hyperplasia that's happening at a basal level. And infection is able to drive differences as well, or this difference is maintained during infection. So there is an add-on effect, but it's not synergistic. It is just an add-on effect of infection there. TRPV1 was previously reported to be expressed on intestinal epithelial cells, and it was reported in at least a few different cancer cell lines and cancer models that it could reduce proliferation. Again, we saw increased proliferation in our TRPV1 knockout in our wild type mouse. We didn't see a basal increase in proliferation by measuring Ki-67. So although we had increased crypt hyperplasia just by measuring the crypts, when we actually looked at Ki-67 in the tissues, we didn't find evidence of increased proliferation. We also did colonic organoids from both wild type and the knockout, the TRPV1 knockout mouse, and looked at EDU uptake indicative of proliferation. And we didn't see any difference there as well. I'm not showing you that data right now. And so what we thought then, or how we took this, or what we took this to mean was that this is really just about the bacteria are driving this. The increased amounts of bacteria that we see in the TRPV1 knockout mice are driving increased proliferation by Ki-67, and that's what's being summarized here, compared to our wild type mice. So there's an increased proliferation from infection at day 10 in a wild type mouse and in a TRPV1 knockout mouse. But again, this is significantly enhanced in the TRPV1 knockout mouse compared to the wild type mouse. And proliferation is back down to normal by day 29 post-infection. So we think that it's really just about how much bacteria are present and that ensuing response from the host, as opposed to there being an intrinsic defect, at least in driving proliferation in intestinal epithelial cells. Well, we also then, of course, like most people on this call, did flow cytometry. And so TRPV1 has also been previously reported beyond CD4 positive T cells. And so we did lamina propria lymphocyte preps. Well, I didn't do them, my student did, and performed flow cytometry as well as intercellular cytokine staining, looking at key cytokines that are known to be important in the host response to Citrobacter. So here, just looking at the total frequency or the frequency of live CD3, CD4 positive T cells, we can see that, yes, and as you would expect at day 10 post-infection, there's an increase in the number of these cells in lamina propria, of T cells in lamina propria. And this holds true as well for TRPV1 deficient animals, that there's no change in the recruitment of those cells. And we start looking, though, at cytokines that are being produced. So again, these are key cytokines known to be important to defense against Citrobacter rodentium. So interferon gamma, positive CD4s by intracellular cytokine staining. We see, yes, infection drives an increase there. There's maybe a slight decrease, but nothing significant. And certainly you wouldn't get excited about IL-17A expression, or production rather. Again, infection is driving massive increases. This is, of course, known. And in the wild type mouse and in the TRPV1 knockout mouse, there is a statistically significant reduction of IL-17 that's present in the TRPV1 knockouts 10 days post-infection. The IL-22, although that has also been demonstrated many times now to be critical to host protection, we don't see a difference, at least in terms of wild type infected versus TRPV1 knockout infected. So with this, most grad students are very, very happy and excited about these types of results. And I thought, well, maybe that's not it. Let's go back and do a little bit more immunophenotyping. And that's exactly what we did. And doing this, what it looks like is happening in the TRPV1 knockout mouse is that there's reduced neutrophil recruitment. So here, what we're showing you is that in our wild type uninfected mouse, you can, of course, find scattering of a few neutrophils that are present, and the quantifications are over here. In our TRPV1 knockout mouse, it's very similar. And if you infect with Citrobacter rodentium and at day 10 post-infection come in and now start enumerating and looking for neutrophils, you can see there's a significant increase that's happening. And this is significantly reduced in the TRPV1 knockout mouse, both by frequency and by absolute number of neutrophils. This is significantly reduced at 10 days post-infection. So I'm not going to walk through all of this, but what we started thinking, well, how could this possibly be? What is TRPV1 controlling? And so we thought of some of these things like, looking at different chemokines, and long story short, or at least as one bullet point there, there's no difference in the sort of usual suspect type chemokines. We also thought of, well, maybe it's something about progenitors of neutrophils, their development, their maturation, and there's no deficits, no defects. Those neutrophils are perfectly normal and perfectly fine, at least by how we can or how we determine them. And so what we thought again is, well, maybe it's about how they're getting in. And so what we did in this case again is look at in the colon, creating a single cell suspension, and then doing staining. This time focusing instead of on laminar propria lymphocytes to actually look at stromal cell compartments. And so gating on blood endothelial cells. So these are 45 live, 45 negative, singlets, of course, CD31 positive, GP38 negative, also known as protoplanin. What we can see is that, yes, infection is driving a significant increase in the amount of ICAM1 on the blood endothelial cell surface. So this adhesion molecule, general adhesion molecule, right, that is going to allow for immune cell binding and eventual egress into the actual inflamed tissue. And this is significantly reduced in our TRPV1 knockout mouse. VCAM as well is also significantly reduced in our TRPV1 knockout mouse that's infected 10 days post-infection was when we're monitoring this. Looking again at MADCAM1, we don't see any real difference here at the protein level on those endothelial cells, and this is not different compared to comparing wild type to TRPV1 knockout. So a nice internal control, at least, in that it's not just every adhesion molecule. It seems to be that there's specific adhesion molecules that seem to be regulated here. And so this is what we proposed as a potential mechanism or a mechanism to explain how in TRPV1 knockout mice that there was a significant reduction in the number of neutrophils that were present. Okay, so to summarize, there are, at least this first part, is that there are many different neuroimmune circuits and components of these circuits. They can participate in lots of different interactions. And again, just so that just because we identify some of these things in one context, be it an anti-inflammatory cascade in endotoxemia, or maybe a model of rheumatoid arthritis, those same cells may actually participate in slightly different ways in a host pathogen context, right? So think of the chat GFP story that I told you about, for example. And this host response then to an enteric bacterial pathogen is coordinated by multiple factors from TRPV1 positive neurons or potentially other TRPV1 positive cells. We don't think it's just other TRPV1 positive cells, and I'm happy to answer any of those questions. And so we think TRPV1 then aids the coordination of a host immune response. And really it's about regulating of an innate immune cell recruitment. But it gets to the question of, well, when you deplete these, why do we have a different effect? And is it just the neuropeptides that are produced? And what would these potentially be? So at the end of this paper, this is what we sort of got to. And it led us to think about, well, what are the major neuropeptides there? Well, substance P has been known for a long time. It's maybe one of the oldest known and established neurotransmitters. And this is encoded by the gene TAC1. And this can be produced by sensory nociceptive neurons. So that's what's being depicted here. This is, again, TRPV1 positive. And it activates a target cell through the substance P receptor. So this is encoded by TAC-R1. And you'll see this abbreviation throughout. This is why I'm pointing it out. And substance P has actually a history of immunoregulation, at least in the gut. And that substance P receptor antagonists and antagonism of that receptor, whether or not it's small molecule pharmacological antagonists or other, have been shown to reduce inflammation in dextran sodium sulfate model and trinitrobenzoic sulfonate. I'm probably messing that up. Induced colitis model in both of these papers. There's also a little bit of controversy with substance P in that other roles, in fact, counter roles, anti-inflammatory roles have been also identified. But this is, interestingly, I think, and it will be important later on in my talk, that substance P could actually be anti-inflammatory as well, depending on context. So there's at least a history or a background of this particular neuropeptide having a ability to drive or modulate immune responses in the gut, not in intestinal inflammation so far. So that's exactly what we decided that we would do. And again, we're using our Citrobacter rodentium model. And in this particular case, what we're going to do, or what we did, is use a very old but still very selective and highly efficacious substance P receptor antagonist. So this is CP96345. And we have used others, but I'm going to talk mostly about this data. And the way that we started doing this, again, so we're going to use our Citrobacter, and we're going to control gavage with LV broth. And at the same time, well, actually a couple hours later, we're going to gavage those animals with either the substance P receptor antagonist or vehicle control. And we're going to do this every day, and depending on when we're taking those animals down. So if they're being euthanized on day 10 or euthanized on day 30, they're getting a gavage of this particular drug. And then when they're euthanized, we're again looking at our normal parameters: colonic hyperplasia, immunopathology, bacterial burden, post-immune response through many different ways. Okay, so the first piece of data that I'm going to share with you, and this is now all unpublished work, is that if we gavage this particular drug and we look at bacterial burden starting at day 3, so the lighter colored or gray line in this particular curve is our CP treated animal, or substance P receptor antagonist treated animal, and this is our vehicle control in the black line. And you can see that there's actually a significant decrease in the amount of bacteria that's present in the feces that's being shed. I'd also like you to realize or to recognize that this is a log plot, so it's actually pretty substantial, some of these differences. When we look just at day 10, so these are different cohorts now, we can see again CP, so substance P receptor antagonism is significantly reducing the number of bacteria in the feces. And if we look on the colon associated, we can see again the same trend, or same statistically significant difference rather, that substance P receptor antagonism reduces the amount of Citrobacter rodentium that's associated with the intestine. Looking at chronic immunopathology or histopathology, so we see CP doesn't actually do anything to non-infected animals. Their crypt length or crypt depth looks pretty much the same. And again, Citrobacter rodentium induces a significant increase in crypt length, suggesting again that there's hyperplasia that's ongoing as a cause of that. And pretreatment substance P receptor antagonist significantly reduced this. Right, there's a significant reduction in the colonic hyperplasia that's seen. Is it really colonic hyperplasia? Well, we looked at Ki67, so again, indicative of proliferating cells, and looking at epithelial cells, so DAPI positive, CDH1 positive cells by confocal. So we've removed the CDH1 channel. It's here. That's what we're showing in this little inset. So yes, obviously, mice that are uninfected and vehicle treated have epithelial cells that are proliferating. Citrobacter rodentium induces epithelial cell proliferation, right? We can see this fairly nicely in this particular micrograph here, 10 days post-infection. In the vehicle treated, but in the CP treated, there's significantly less Ki67 positive cells that are there. So this drug is preventing infection-induced IEC proliferation or crypt hyperplasia. And then we still have a increase at day 29 in the number of proliferating epithelial cells. And this actually now isn't different with CP treatment. And that's what's quantified and being shown here. Okay, so probably like many of you, we had a lot of questions about this. We thought, well, we're gavaging this. Are we just doing something to the bacteria directly, to the pathogen directly? And we don't think that's the case. And one of the reasons why we don't think that's the case is that when we use a different drug, so this is a slightly newer drug, and we inject this IP instead of by giving it by oral gavage, we still have this significant difference in the number of bacteria in the feces. This is looking at just day 10 post-infection. And if we look at the colon and enumerate the bacteria that are there, again, now this SR14033 is also significantly reducing the amount of Citrobacter rodentium that's in the colon as well. Oh, and this is getting cut off. I'm sorry. This is basically just saying CFU per mil, and this is showing up. It's a log plot. We asked if CP96345, the first substance P receptor antagonist that we used, was potentially bacteriostatic or bactericidal. Here we're just showing growth curves where we actually plated out the bugs. So we didn't just do OD. And we saw that there was really no difference in any of these doses compared to vehicle control, just DMSO, effect on Citrobacter growing in vitro. We also did bactericidal assays, so using a flow-based kit, and found that the bacteria were just as viable, at least by that assay. And we've also been looking at virulence gene expression that you can induce in vitro by growing the bugs in DMEM. And have not found any change. So long story short, essentially, is that we don't think this is just about the substance P receptor antagonist impinging upon or directly killing or reducing virulence gene expression. It's not that simple. That we think there's other effects, and we think it's actually on the host. But what about the host? Well, it's also been known that substance P receptor signaling on blood vessels can cause blood vessel dilation. And we thought, well, maybe there's a role for oxygenation or nutrients or something of that sort. And so we thought we'd tackle the oxygenation portion. And to do this, sorry, I'm just getting ahead of myself. The oxygenation is absolutely critical to Citrobacter rodentium infection. So when Citrobacter infects mice, normally the intestinal epithelial cells are held at a almost hypoxic state. So this is the work by Chris Lopez and Andreas Baumler's lab, who's actually at UC Davis. And what they showed is, so you have this very close to hypoxic state in some of the intestinal epithelial cells, especially close to the lumen. And so CP9, so using this particular approach, we looked at what happened with our substance P receptor antagonist, and there's really no effect, at least in an unaffected animal. In an infected animal, there's this complete change in metabolism. The red that you're seeing here is actually Citrobacter rodentium. So it's absorbing this hydroxy or hypoxy probe. But the intestinal epithelial cells, I think hopefully you can see, there's no red associated with it. So they're not under a hypoxic condition. And CP96354, our substance P receptor antagonist, didn't change this either. So we don't think it's just about changing of oxygen balance and sort of this rewiring of metabolic pathways that are actually beneficial to the pathogen. That's not what CP is preventing from happening. So what is it then? So of course we go back to looking at T cells, as most immunologists probably would. And so I'm showing you confocal, and I'll show you flow cytometry in a minute. And so we took sections from our different animals. So here's our vehicle treat. You can see Citrobacter rodentium induced T cell recruitment as we expect. And these are staying quite high. Lots of T cells are still there at day 29 post-infection. Our CP96345, so our substance P receptor antagonist treated animals, there's no real increase in recruitment. In fact, it's significantly reduced compared to wild type. That's what's being shown vehicle treated. That's what I'm showing over here in these bar graphs. So there's significant reductions, and matching what's on the in the micrograph, you can see that there's still this significant reduction even at day 29. So it's reducing the amount of T cells that are coming in, are being recruited during infection. And so we thought, well, let's look at some cytokine or cytokine mRNA and some other host protective factors and try to get a broader view of what's happening here in the lamina propria, or at least in the full colon, full thickness colon section. First, looking at interferon gamma, again, at 10 days post-infection, we see that CP96345, so substance P receptor antagonism, significantly reduces the amount of interferon gamma mRNA, and this is the same at day 29. Looking at things like IL-17 or IL-22, we don't see real changes, or at least for IL-17, we don't see changes with substance P receptor antagonism at day 10. But there is some difference, or a fairly large difference rather, at day 29. And again, IL-22, at least by mRNA, doesn't really seem to be statistically impacted. Maybe there's trends, but it's certainly not statistically significant. The ramifications for the host, though, are, I think, pretty important and interesting. So looking at inducible nitric oxide synthase, so encoded by NOS2 gene, looking for transcripts of this, we see at day 10, there's significant reductions in NOS2 expression by RNA. And this holds true as well to day 29 post-infection in substance P receptor treated animals compared to vehicle control. Other factors like Reg3 gamma, again, that there's upregulation during infection, substance P receptor. So it seems to reduce this, but it's not statistically significant at day 10 and only reaches statistical significance at day 29. So there's key aspects of the host response that are being altered, not just that T cells recruitment are different, but that the downstream effects of that are in fact being impinged or inhibited or blocked as well. Looking at flow cytometry and of lamina propria lymphocytes, we see that there's actually reduction of the amount of T cells that are coming in our infected substance P receptor antagonist treated mice compared to our vehicle control infected animals. So you can see there's a significant difference there. And when we look at what are those cells making, the CD4 positive T cells, we can see that there's a lot of these cells, or a good proportion of these cells, are making interferon gamma in our vehicle-treated infected animals, and this is significantly reduced in our substance P receptor antagonist-treated group. Here, at least for protein, we can see that yes, there's IL-17 producing T cells in our vehicle-infected animals that are present in lamina propria, and this is significantly reduced. The amount of protein detected by flow cytometry is significantly reduced in those substance P receptor antagonist treated animals. In IL-22, there's really no difference. We see increases, infection-induced increases, but it's not different between vehicle and antagonist treated. We also looked at whether or not FOXP3, so regulatory T cells, were going to be changed, and we found no difference whatsoever in chronic lamina propria or in mesenteric lymph nodes or in spleen. So we don't think that substance P receptor antagonism is altering a Treg population. So again, I was thinking, how does this actually happen? We went back to doing flow on our stromal cell populations and looking for ICAM, VCAM, and MADCAM expression. And you can see that infection is inducing expression of these different populations. It seems like significant stars disappeared from here. I'm sorry, I don't know why. So infection-induced expression of these on blood endothelial cells, both for ICAM and for VCAM. This is not statistically different here. But when we look at MADCAM, we can see that the infection-induced increases in MADCAM expression on those blood endothelial cells in vehicle-treated mice, it's significantly reduced in our substance P receptor antagonist treated. And so we think this is the mechanism, or one of the mechanisms, for why there's less T cells that are coming in to the colon or being recruited to the colon in those Citrobacter infected animals treated with that drug. Okay, so we then started to think of what would happen to the actual T cells and whether or not this could be a direct effect on those T cells, on proliferation or other things. And the way we did this is a little bit different. We decided not to try and look for Citrobacter specific effects, but what we did was to use OT2 positive T cells, so T cells, TCR transgenic T cells, are specific for the epitope from ova. And we treated animals with vehicle or our substance P receptor antagonist daily. We IV injected T cells, and then we gavaged ovalbumin. The following day, the T cells that we injected were labeled with a cell trace proliferation dye indicator, so we could find them and actually see how many divisions that they underwent. And then we harvested the mesenteric lymph nodes and looked at proliferation in either our vehicle-treated or lymph nodes from our vehicle-treated animal or our substance P receptor antagonist. We didn't see any difference there. We didn't see differences in either division or just if you looked at proliferation. But we did notice that there was actually statistically significant increases in the amount of the actual numbers of these T cells that are in the mesenteric lymph nodes. And so we don't know yet, but we think that this might suggest there's some sort of retention or that trafficking is different. And we also looked at dendritic cell populations here because that could be another reason, of course, why citro is, or the response to citrobacter is different. But we found no difference in dendritic cell populations making it to the mesenteric lymph node. And these data, at least to us, argue that DCs are capable of picking up an oral antigen, orally delivered antigen, and running back to lymph nodes and presenting that antigen to T cells. And so we think that it's really just the substance P receptor antagonists, at least in Citrobacter, are preventing T cell infiltration or recruitment into the colon, as opposed to being a broad proliferative defect or defect in the cells. Well, that's nice to think about that way, but we thought we needed to prove this a little bit better. And so the way that we did this is to make a substance P receptor, so TAC1R conditional, a T cell conditional knockout mouse. So we had actually during the height of the pandemic, since we had started that process just before, started making these mice, and luckily they were able to continue during the pandemic. And so we have these TAC-R1 flox mice, and so LCK Cre negative TAC-R1 flox are of course wild type, and the LCK Cre TAC-R1 flox are T cell conditional knockouts for substance P receptor. So we've run them through this exact same pipeline and model system that I've told you about already, and looked at these different outputs. So I'm not going to show you all the data from this, but what I think is the most interesting, personally, in this particular case. So here's our wild type in the white over here. And now we're just looking in lamina-propria lymphocytes. We're looking at percent of live, frequency of live CD3 positive, CD4 positive T cells. And you can see infection causes an increased number of these in the lamina-propria in the wild type. And this is similarly increased in our T cell substance P receptor conditional knockout mouse. So there's no effect on T cell recruitment if you just knock out the receptor on those T cells. When we start looking at what cytokines, again, that these particular cells are making, we can see that interferon gamma is actually the cytokine that's most impacted. So infection is driving recruitment of interferon gamma producing CD4 positive T cells in the wild type. And there are some, of course, there is an increase, but it's significantly decreased in our substance P receptor T cell conditional knockout mouse compared to wild type control, but with infection. So the substance P receptor seems to be doing something that is allowing for or increased recruitment or production of interferon gamma. And we thought, well, a good way of discerning this, we took these cells, of course, in vitro and did differentiation assays, and we could find that they were equally capable as the wild types were in making or becoming Th1 type cells, making interferon gamma in vitro. So we don't think it's that they're not capable of becoming that T helper subtype. It's that there's something else at play within this system. And again, looking at other host protective cytokines like IL-17 or IL-22, we don't see differences between our wild type and our conditional knockout mouse during infected conditions. Okay, so to summarize what I've just told you, blockade of substance P receptor, it reduces bacterial burden, and this can reduce immunopathology. This is mostly crypt hyperplasia and the proliferation of intestinal epithelial cells. And this also reduces our pro-inflammatory cytokine production within the colon. And this is, at least in part, a large part, due to recruitment of T cells. Our substance P receptor T cell conditional knockout mice reveal that there's T cell intrinsic effects. There's reduced interferon gamma, but perhaps there's other cells that are responding to substance P or other molecules that can signal through substance P receptor. And this, I think, is really shown through our data that there's no change in T cell recruitment in our T cell conditional knockout mice. There are other cells that are responding to the signaling. So I think that understanding all of this really could allow for selective activation of this pathway, or maybe even inhibition of inflammation in the gut. That substance P receptor then could be used to fine-tune immune responses during enteric bacterial infection. And I think, again, it re-highlights this idea that there are lots of different neuroimmune circuits, and the cells that are making those circuits up, they're tuned by evolution to deal with many different threats, and maybe we shouldn't be surprised then that they could have lots of different potential roles depending on a different threat. I also think we might need to, as a community, move away from this idea of, when I say circuits, I think it evokes in people's minds this idea of electronic circuits with fixed or static parts. But these are cells, right? These are not copper traces on a circuit board. It's not like your phone or your laptop with a set piece or a set number of parts that all respond in the same way. The cells that are experiencing inflammation are going to change as any cell would. And so I think we have to appreciate that going forward, that when we identify these things in healthy animals, these neuroimmune circuits in healthy animals, that's only part of the picture. We need to understand how inflammation impacts that actual neuroimmune circuit as well, that it can't just be a defined thing that we don't ever change. And so again, I'd like to thank all the people that contributed to this work, Michael Kremen, especially Valerie Ramirez, and our core facilities, our collaborating lab, the Grow Lab, and of course, funding from NIH. This just wouldn't be possible without them. So thank you, and I'm happy to take any questions that you may have.
Okay, great.
Recording stopped.
Thank you so much, Colin, for this wonderful talk and truly inspiring. So now the floor is open for questions. If you have any questions for Colin, please either type in your question in the chat box or just raise your hand and directly ask a question. So I see there's already one question in the chat box came from Yashi Jen. A nice talk. CP96345 could reduce immune cell recruitment and the pathogen burden. So how does CP accelerate the clearance of pathogens?
Yeah, no, that's an amazing question. And I think you can tell we're still thinking about this. Our current thought process on this is that, and this will be controversial, so I like this a lot, actually. I think we've, as immunologists, have become in this sort of silo of all immune responses are good and that it's bad for the pathogen. And I think evolution is better than that. And I think the pathogens have evolved under this constant threat, right? And I have no data for this, so I'll say that first off, or should have. And I think what we might be starting to see is where some pathogens are actually adapting or using this. So it's almost like a judo kind of thing, right? That our strength could be their strength, that they might use or subvert some of these what we think of as being protective processes to actually enhance their survival. And so if we can tamp down certain aspects of that inflammation, we're now removing the ability for them to take advantage of that. That's how we're thinking of it, but I don't know if we're right, and I have no data to support it right now, but that's what we're thinking. That's a great question, and I wish I had a better answer for you.
Okay, great. So next question comes from Ping. Very nice talk. Would this be the same situation for the fungi infection? And have you looked at other cells like neutrophils, macrophages, epithelial cell change? And have you looked at any change in the pair patch in the intestine after the infection?
Yeah. Um, Fungi we have never done. So yeah, almost all of our pathogen work is enteric bacterial pathogens. I think that's an amazing idea. I have no idea if that would look the same or not, but that's a really good idea. We've looked a little bit at other cell types, depending on what model or what we're ablating. Um, we are doing things. So substance P receptor has been shown to be on intestinal epithelial cells. I think I mentioned that before. So we're doing things like a Vil and Cree cross to say, is there a role at all for substance P receptor on intestinal epithelial cell? We actually can't find a lot of substance P receptor on intestinal epithelial cells. So if we sort, we do an EDTA shake, isolate those epithelial cells, in fact, sort away immune cells. We don't really find a lot of mRNA for that. It doesn't rule out there's a rare population of IECs, subpopulation of IECs. But we haven't really looked at epithelial cells yet. That's in I think it's the next 2 weeks that those experiments will be done. Neutrophils, I think we have found some substance P receptor on there. I don't think anyone really knows what it does, and it doesn't seem to, at least in our hands, to be screaming like there's not a high level expression that we've seen. But yeah, I think these are great questions and great points. I wish I had a better answer for you. Pairs patches, I think that was the last part, pairs patches. We have not looked specifically there, and that's mostly a function of Citro doesn't infect, or at least in our hands, doesn't infect small intestines. So I don't know how that would work. I mean, Isaac Chow has in their group, that's the Nicole, I think it's Nicole is her first name. And Cell in 2020 looks at salmonella infection and invasion in pairs patches and how CGRP from sensory nociceptors are controlling development there. But we have not done any pairs patch work.
Okay, great. So I also actually had a question on the first part of the talk. Does the bacterial infection induce chat expression in T cells? Or I guess a more general question is, what regulates the chat expression in T cells?
That is a great question. So in that particular context, Citrobacter infection, we didn't look at, was it the Citro itself regulating, driving increased expression, or is it recruiting? We think it's recruiting. We do think it's something specific to the bacteria. And why I say that is when we did DSS colitis in that same paper, we didn't see, even though we could see more T cells, and it's not really a T cell heavy model. It's not a T cell centric model, right? DSS colitis is mostly neutrophil driven. We didn't see increases in chat in those T cells. So prior paper, when I was a postdoc in TAC Max lab, we looked at what was required for chat expression in T cells. And in fact, some people on that lab have continued. So Maureen Cox as well. So she looked at CD8 T cells. We looked at B cells and CD4 positive T cells. And definitely TLR signaling, MYD88, and the microbiome are key in driving chat expression in lymphocytes. Absolutely key. If you knock out MYD88, significant reductions in chat positive T cells and B cells. Excuse me. If you ablate microbiome by doing a nasty cocktail of antibiotics and or significant reductions as well. So there's definitely something coming from the commensal microbiome. It looks like it's a PAMP of some sort. I don't know if it's one particular or if it's many. You can stimulate with LPS and things like that and drive this as well. But we didn't get to a fine sort of, ligand pool and what of that ligand pool would actually drive in that. So amazing questions. And I think it highlights for everyone still on here that there's a lot still to learn about this. And it's a whole new area,
Absolutely,
honestly.
absolutely. It's, I
Still.
think, really fascinating. I guess a follow-up question would be, does the chat expression affect any of the, let's say, the cytotoxicity or memory phenotype of the T cell.
Yeah, that's Maureen, absolutely. And so that's Maureen Cox's follow-up paper. It's Science 20, oh, I'm gonna forget the year, 2018, I think. Sorry, Maureen, if I got that wrong. Um, and yes, exactly that. So in that paper, I believe they showed that I'm trying to think of if it's also follow-on stuff that she's been doing because she's still continuing to work in this. So yes, absolutely, chat expression aids clearance of LCMV, and I'm not an LCMV person, so I'm gonna stop before I put my foot in my mouth or mess this up. But absolutely, and it was important in viral clearance and actually having the T cells, cytotoxic T cells, gain access to viral reservoirs in different tissues. So absolutely, and again, I think that's a fascinating area. Naive CD8s. If you take that ChatGPT mouse and just do flow on spleen, there's less than 0.5% of CD8s will be chat positive. And when Maureen infected those animals, it went screaming. So there is definitely something else going on, and it's in terms of how chat's regulated. And I think that's also really fascinating. So I'm excited to see what comes out of her lab, or is continuing to
Cool.
come out of her lab.
Great, great. Thank you so much. So have another question came from
Mhm.
LWY. Very good job. Whether TAC1 or knockout in T cells show protection role in the clearance of pathogens. Which cells mainly express the substance P receptor?
Yeah, no,
TAC1.
that's a fantastic question. We don't truly know in terms of so we know T cells. So if you take lamina propria lymphocytes and you sort CD4 positive T cells, right? And you look at so there's no good antibodies, or at least we found no good antibodies that are knockout validated or biologically negative validated for substance P receptors. So you have to do a lot of this with mRNA, which it has its own issues, and that doesn't faithfully recapitulate protein and all of that, as many of you are aware, or all of you are aware. When we've done these sorts and then done qPCR, looking at mRNA expression, it's definitely T cell population has a low level of substance P receptor. And when we look at the same cells from now sorted from an infected mouse, that expression goes up 400-fold. So there's something about activation. There's something, whether it's a specific, is it a T helper one, that we don't know that yet. We really don't know that yet. But it is a great question. For sure, it's T cells. We've seen some other immune cells, and we've seen blood endothelial cells, as would be predicted from single cell seq databases and things like that. But yeah, we don't know if it's constrained to just Th1 or if it's Th17 or if it's not really a T helper subtype restricted thing at this point. It's a great question.
Okay, great. So if there's no more question, let's thank Colin again for this wonderful talk and also a really nice discussion. And thank you so much, Colin, for joining us today to share with your really exciting work.
Oh, thank you. No, it was fantastic, and thank you to everyone in the audience for asking such amazing questions. Really, it was a lot of fun.
Okay, great. Have a wonderful night and see you
Great.
next time.
Thank you, everybody. Bye.
Bye.